Okay, I'm going to make this as simple as I can possibly make it. It's a little bit complicated, but you're fully capable of understanding it top to bottom. Everybody understands that in your DNA, it wraps around like this, and it's little bars, boom, boom, boom. And every bar has 4 codes. And that's a gene. Just consider that 4 boxes a gene. All right? And if that gene is in a certain condition, in a certain order, and they're CTAGs, and if the C's go with the G's and the T's go with the A's, and they cannot intermix, but they can reorder themselves. And depending upon the order of it, it says, is this gene turned on, yes or no? Basically, that's how I'm going to approach it. Now, once it's turned on, yes or no, it has to express. And what does that mean? That means it has to go out and get somebody to help it express itself, to do its job. And what is its job? Its job is normally to create a protein. And what is a protein? A protein is a chemistry set. Literally, it's a chemistry set. And what it is, normally I'm going to think of it this way, and I think it works quite well this way. Bacteria that are in your body, in your microbiome, your gut, are creating all kinds of chemistry that these genes call on. They're called enzymes. And what they do is they break up the molecules, and instead of having a bunch of this ball of stuff over here, they need 6 of these and 5 of those and 4 of these and 2 of these. And they call on the bacteria. They say, here's what we need. We need an enzyme. We need this kind of enzyme, that kind of enzyme. And what they're talking to is lactobacillus and prevatellas and all these different types of E. coli's and all those. They all make products. They don't just lay there, but they don't just continuously squirt those products out. They have to be turned on. The gene expresses itself by turning on the bacteria. The bacteria break the chemistry up. The chemistry comes down and does exactly what it should do to fight cancer, to break the cells, or to block the pathways, or to do the biology required to keep you fit. Now, here's what I have come up with. And I think it's damn accurate, because everything I am finding, and you see that? I think maybe you can. It's all gastrointestinal issues, virtually 100%. Now, this morning I'm looking up something. I don't even know how I got into it, but the guy was talking about a hard time to understand how a gastrointestinal issue could either create these 3 different things, and it seems to be all in the same gene. It's turned on or off, or they're cutting into these genes and they're splicing. And some people have the gene in a certain way, some people have it in a different way. If you have it in one way, you may get Crohn's, Parkinson's, or schizophrenia. If you have it in the other way, you don't get those. Well, I know that's your gut. That's your gut. That's your gut. All of these things are from gastrointestinal issues. If you have that gene in a certain condition in your body, because some people get them. They get them this way and they get them that way. And, I remember how I got into this, because they're editing babies. They're editing babies and doing all kinds of things. And this is about the microglia, but where was it? That's the mice. This is, well, let me find out let me get back to you. All right, I was digging around looking into the autism and the gut microbes, because it's very, very well linked now. Virtually everybody understands that now. Now, this guy, Joe Scott, very good guy. Somebody tipped me off to him, and this goes back a couple of years ago, and no, last year, I guess. And this was about these twins that they spliced genes on them. You know, this is like, it's like Babylonian stuff, where they spliced the genes on these kids to make it so they couldn't catch HIV. Now, apparently they found out that there's some form of a pathway that if the gene is this way, the pathway's open. If it's that way, the pathway's closed. If the pathway's closed, you can't get HIV. Now, some people have the pathway closed already. Just come out of the box just like that. The other ones, it's open. And then you can get HIV. I guess everybody can't get HIV. This is what I'm taking from this. Now, and this guy was very, very good about how he presents this. But the point being is that they went ahead and altered the genes on these kids. They went in and they spliced their genes and cut them in there before they started to grow. And if they get it on the wrong side of the cell division completion, and they don't know how long it takes to complete. They don't know when it's complete. You grow. Cell division doesn't necessarily have to all of a sudden say, okay, I'm going to be an eyeball. Instantaneously, it's an eyeball. It may have to go through some changes. Changes, yeah. How do you know that? Who's going to say yes? Who's going to say no? These kids are Dr. Frankenstein experiment. Nobody even knew about this apparently, and they just went ahead and did it. This is a good guy to look into. He's good. Joe Scott, The Chinese Genetic Experiments That Shocked the World. Answers with Joe. I got a lot of videos. I was looking at some of the stuff he does. I like it, and he seems like a pretty good thinking guy. And we need more of that. I'm going to tell you that right now. And I'd like to see a father of dragons there. They're all real. They're showing satellites now. Those are the father of dragons. I don't know. I think the dragons were the father of dragons, Joe. We should talk, brother. All right. Let's look a little deeper. All right. So the CRISPR is how they cut and splice the genes. And they're talking about, this is a bombshell. Nobody expected this. Well, I guess somebody expected it. But here we go back to February of 2019. The twins might have had their brains, what? Let's see. I haven't looked at this yet. But they might have had their brains scrambled. Inadvertently enhanced. Well, oh boy. Isn't that something? New research suggests a controversial gene editing experiment to make children resistant to HIV may also have enhanced their ability to learn and form memories. Oh boy, there's going to be a rush to do that. I'll tell you, this is not good. These kind of things, in my mind, not good at all. It says new research shows the same alteration introduced into the girl's DNA, deletion of a gene called CCR5, not only makes mice smarter, also improves human brain recovery after a stroke. Is that all? Is that excellent? Is that the only thing it does? All right, I've seen mostly, this is all good, and it triggered all these good things to them. Well, this is just from a few months back. This is December 2019. It's February 2020 right now. So, and it's saying more than a year prior to this, excuse me, the world was shocked by these Chinese biophysicists, He Zhonghui's attempt to CRISPR technology. That's, like I said, they spliced the genes to modify human embryos, make resistant to HIV, which led to the birth of twins Lulu and Nana. Okay, so now, crucial details have been revealed in the recent release of excerpts from the study, which have triggered a series of concerns about how Lulu and Nana's genome was modified. All right, so here's how the CRISPR works. CRISPR is a technique that allows scientists to make precise edits to any DNA by altering its sequence. The CTAGs, you click this and move that one over there and take out a piece here and insert a piece there that is in a different sequence. Now, so when using CRISPR, you may be trying to knock out a gene by rendering it inactive, or trying to activate or achieve specific modifications, such as introducing or removing a desired piece of the DNA. Because the DNA either turns it on or turns it off. They express or they don't express. And that means that you either have the ability in your body to deal with something, or you don't. Some things will allow you to get invaded when other sequences say this pathway is not available. And that's what's happening with HIV. Apparently in our bodies there is a pathway that says, okay, if you come down this road here, you can come in and give me HIV. And if I say this road isn't here anymore, which is what the sequence does by removing this or changing it, it says, oh, you can't come here anymore, so you can't get HIV. Now, when using CRISPR, you may be trying to knock one out or put one in. So gene editing with CRISPR relies on association of two proteins. One of these proteins, called Cas9, is responsible for cutting the DNA. So they at a certain place. The other protein is a short RNA molecule. That's a little gene code, which works as the guide that brings the Cas9 to the position where it is supposed to cut. Well, all right, so it's one of them is a tag that brings it to the place that cuts it, and then it cuts it there. The system also needs help from the cells being edited. DNA damage is frequent, so cells regularly have to repair the DNA lesions. The associated repair mechanisms are what introduce the deletions, insertions, or modifications when performing gene editing. So you're using your own, the person's own body system, apparently, to work on this DNA. And how the genomes of Lulu and Nana were modified. Okay, Janucci and his colleagues were targeting a gene called CCR5, which is necessary for the HIV virus to enter into white blood cells and infect our body. All right. On the white blood cells, the lymphocytes, apparently there's some structure on there. It's topography. It's literally hills and valleys and hooks and so forth, that this thing comes along and it grabs in there and says, ooh, this fits just right, and it stays there. Then you can get infected. If it comes along and it just can't hook, it just keeps going, well, apparently that's what this does. It changes the topography of this particular site. So one variant of CCR5 is the delta 32, missing a particular string of 32 letters of DNA code. This variant naturally occurs in human population and results in a high level of resistance to the most common HIV virus. So it happens naturally in the population, apparently. So they want to switch it off. Now, Janucci's team wanted to recreate this mutation, and it's a mutation, so it's not just, apparently this doesn't change on and off. You either have it or you don't. Now, other things in the DNA turn on and off, and they do it seasonally. We're going to probably discuss that a little later. Now, because I wanted to get into, a lot of this about how these genes really work in your body. I'm not sure they really understand some of the intricacies. And I may not either, but I want to discuss what I do think I understand, and I'd like to have somebody address what I think. All right, now, so as a result, they generated different mutations of which the effects are unknown. So they modified these CCR5 close to the delta 32 mutation. Apparently they can't just say, okay, make it a delta 32. They make it, I can see those molecules are sort of lining up the same way, close enough. So as the result, it generated different mutations, not the exact copy, of which the effects are unknown. They don't know what's going to happen. It may or may not confer HIV resistance. It may or may not have other consequences. Right now, it's a rogue program that's running around in your body. It's a rogue program. And that's because everything you have that consists of DNA is a program. That's all it is, a program. If it's this way, it makes something happen. If it's this way, it makes something else happen. There's a lot of different little bits and pieces to it, but in essence, it's either on or it's off. And once it's off, it shouldn't affect you. Once it's on, you should have the ability to make that thing express itself and do its job to protect you. But the only way it can protect you is to have bacteria that it can work with, because the bacteria creates the chemistry. I'm telling you, and you look it up, you can find out your immune system is dependent upon non-human bacteria. Look it up. So now, as a result, we don't know what's going to happen with these things. Worryingly, they did not test any of this. They just went ahead and did it and see what's going to happen to these kids. Let's see what they do. They went ahead and implanted the embryos. Really, that's a hard thing to justify. You know, somebody's going to have to deal with somebody at the end of their life. Because I'm going to tell you right now, I'm looking at there's more to this life than this life. I'll leave it at that. The mosaic effect. A second source of errors could have been that the editing was not perfectly efficient. This means that not all cells in the embryos were necessarily edited. So some of them had this edit and some of them had the other edit. Well, what does that mean? They're conflicting. There's going to be some source of a problem. So, and it says when an organism has a mixture of edited and unedited cells, it's called a mosaic. While the available data are still limited, it seems that both Lulu and Nana are mosaic. Oh boy. This makes it even less likely that the gene editing babies would be resistant to HIV infection. The risk of mosaicism should have been another reason not to implant these embryos. Moreover, editing can be unintended impacts elsewhere in the genome. So not only is it affecting whatever they're trying to affect, they may be turning the guy's eyes sideways, or who knows what they're doing. All right, when designing a CRISPR experiment, you choose the guide RNA so that the sequence is unique to the gene you're targeting. However, off-target cuts can still happen elsewhere in the genome at places that have a similar sequence. Not only that, this is all new. They have no idea what some other. Let me put it this way. I was in the software business. We had extremely organic programs. They did credit card readers and cash drawers and scales and you know, video monitors, and all these things were integrated, and they worked with push of a button, and it was all integrated. Now, in our programs, whenever you changed a program, you had to change it from a source code, and you had to know the source code. You had to be able to edit that source code, because guess what? When that source code sent out its information, it sent it out with what's called, oh boy, I can't read. It's a number that it generates. And that number is, I'm going to remember the name, don't worry, it's a long time. It's been 20 years since I was deep into that, but what it is, it's a handshake. It says, okay, here's all the stuff I sent you. So an A is a 41, a B is a 42, a O is a 86, and it goes, and all these numbers add up in a register over here. And then it says, what's the number here? It says it's 1052. It comes in hexadecimal. So there's a D257. That's all it is. It's going to be a number like that. And then on the other says, what do you got? And he says, well, when I added them all together, I got a D257. It's okay. That's a handshake. We're good to go. Well, guess what? If you change it here and you send it out, and that guy's over there looking for D257, because that is supposed to be the code you send, and you get a D259, he says, whoa, we got an issue. This is not with me. And he said, well, what are we going to do? He said, I don't know. And then you are in the deep end of the pool. That's all I can tell you. All right, it was a bad idea to begin with. Absolutely. We should stay away from this stuff. This is not our realm. Implications for gene editing as a field. There should be no field. This is not where we should be working. We should be trying to just deal with what we have instead of changing what we have. And this is not going to end well. I can tell you that right now. And when you start to break this down to the bottom line, which in my world is there a creator? Is there a God? Did God create this stuff? Did God create this code? Absolutely. In my world, that's what happened. God created this code as a source code programmer. Do not screw with this code. That's not our realm. You don't go down and say, okay, Sony or whatever, these big Apple, I want to program something. I'm going to just change your codes. You can't do that. Just can't do that. And we're into something that replicates its own self according to a design created by a designer that has nothing to do with us. We are the product of the design. We should stay the hell away from designing it. All right? We've gone way too far to the other side of the rainbows, my friend. And everything that is just something you don't want to deal with, they just don't deal with it. It doesn't matter to anybody anymore, it seems like. Anything that's acceptable is acceptable. Let's just go ahead and do it. Nah, don't worry about it. And we'll worry about it later. And later is going to be later, too late. That's my world. All right? And so I'm just going to say my things that I have to say. And I have been. Nobody's listening, so it doesn't much matter, but that's what I am going to continue to do.